Thank you all for coming today. It's better to be in here than outside right now, so this is a no-brainer. So when I was asked to come up with some topics, I thought, something that was, like Gloria said, across departments, across colleges, schools. I thought, genomic gene information, something like that seems perfect. In 2000, the first draft of the human genome came out, and we finally had the map of what it meant to be human, and we've all been trying to figure out what that means for a while. And then maybe some of you saw earlier this year, the complete draft of the human genome finally came out. The last 8% was finished and all the I's dotted and T's crossed. In that 22 years, there's really been an explosion of companies and technology sort of driven by that endeavor and also sort of drawn into the genomics era that has made genomic information, DNA, affordable, available. And I think we're going to see tonight how it impacts everybody's research. I mean, it's really impacted research, but also society. Like when you reflect now, thinking about this, it was remarkable how many different places you see genomic information come up. Whether it's personalized medicine, people are thinking about their own genome and how it relates to their health, whether it's, the next version of COVID or the new strain of COVID or an emerging pathogen coming up. Forensics, you can't watch a police drama these days without someone doing a DNA swab and sending it to some database. The Nobel Prize was just awarded a few weeks ago to Dr. Svante Pääbo for ancient, genomes. And even more immediately, a few years ago, I got my parents' 23andMe, the genome testing kit, for a Christmas present because I'm a romantic. And so my mom did it. And if you are part of that, you can connect with people on the platform if you're related. And so they were interested in the ancestry part. And she still to this day tells me, oh, another person from Norway contacted me. We're emailing. So it's impacted her life like in a real tangible way. So I'm excited tonight. It'll be fun to see how genomic information is in people's research, but also an opportunity to talk afterwards about what the work we do, how to impact society, and the genomic era is here, so we're going to have to deal with it, and hopefully we can help steer it. With that, I think we'll start with Charles.
Well, thank you for the invitation. It's really nice to occasionally give a talk here at BU. I don't do as much as I used to do. It's also kind of interesting for me to human genome because I'm not doing, 30 years ago, some may know that I was involved in initiating the Human Genome Project, and I'm not doing anything in human genes. So the history, let me just say a few words so I don't run out of time. So most of my career, I did immunology, and more recently, the last 20, 30 years, some cancer genomics. I've been doing data mining all along, and that's all in the past. About 5 years ago, I got interested in climate change. I couldn't believe what I was reading, so I started doing some calculations myself. And what I found was actually a little bit worse than what I thought I would find. You hear a lot about climate deniers, and you hear a lot about Paris Accords, and something has to be done, and everybody's hopeful about the Paris Accords. Well, it didn't take more than a week to realize that there's nothing there. We can meet the Paris Accords. They're not being met. Even if they were being met, we'd go well over 2 degrees centigrade post-industrial. So the serious problem, I'll put this in perspective with just 2 numbers. So that's when I got interested in agrigenomics, because drawing down carbon by traditional agricultural methods goes back a long way, that idea. But using synthetic biology does not, and not many people were looking at that. And here's the problem. There's currently an energy imbalance of 2.3 watts per meter squared. Now, I heard a congressional testimony by someone who was poo-pooing the idea that there's a climate problem. And one of the things he said is, well, we have 250 watts per square meter impinging upon the planet, or 230. This is only 1% of it. How could it have an impact? Well, in fact, you multiply by the number of square meters of surface area, and what you get is the power delivered is the equivalent to 5 Hiroshima bombs. 5 Hiroshima bombs per second energy is being delivered to the planet, most of it being absorbed by the ocean. Huge amount of energy. The current concentration of atmospheric carbon dioxide exceeds pre-industrial levels by 1 trillion tons. It's hard to grasp that number. And how do you get 1 trillion tons out of the atmosphere? In other words, you're going to return to planetary balance we'd have to get 1 trillion tons or some substantial fraction of it out of the atmosphere. The third problem, this is with CO2 and a few other gases, is that CO2 remains in the atmosphere for centuries. And this shows what happens. Just to give you a number, 500 years from now, 20 to 25% of the CO2 that's emitted today will still be in the atmosphere. So and that's just carbon dioxide. I'm not talking about methane, I'm not talking about nitrous oxide. Nitrous oxide also has a long lifetime in the atmosphere. And it's much more efficient in capturing heat. So this is a serious problem. And even if we started today, and that's what I'm going to say a few words about now, this slide is a little bit, there's a lot of information on it. So let me just say very briefly what it means. The lower blue, and the idea here is that every year, something like 120 gigatons of carbon in carbon dioxide is photosynthesized, and about half of that is returned to the atmosphere by trees or by plant life. So 60 gigatons is returned to the atmosphere just by trees. And the idea is to interfere with that step a little bit, to turn the carbon, rather than use it into sugar, to turn it into something that like calcium carbonate, for example. Okay, so what this lower curve shows, the lower blue line shows that if 10% of the planetary trees, and of course that's very difficult to do, but 10% were engineered so that they were 90% efficient, so they only return 10% of the carbon they take in back to the atmosphere, what the result you would get over the next 80 years is shown in that blue line. And something else has to be done. You have to be regulating. You have to be regulating carbon so that by 80 years from now, there's no more emissions. You've gone to zero emissions. So substantial requirements, and you get the blue line. But you have to do both. You have to regulate, and you have to draw down. If you do only one or the other, for example, if you just regulate, where you just go to zero emissions over the course of the next 80 years, that upper red line is what you get, because you're still emitting carbon during this period of time. So it continues to go up. If you just draw down, you get this diamond, this diamond line here. So you've got to do both. What the black line shows is the effect of delay. In other words, if we could implement the strategy immediately, and that's what I'm saying, which you can't do, obviously. So this is like an Alice in Wonderland fantasy. You get the delay of 20 years gives you the black line. So you see you have a substantial loss of opportunity. The more you delay, the more loss of opportunity you get. And that goes non-linearly. That loss goes non-linearly with the delay. Okay, so as I was saying before, I've been working with the Gates Foundation now for the past year and a half. They're very interested in both mitigation and adaptation. We've been pulling together international leaders in synthetic and systems biology who have innovative approaches, both to crop yield. The reason I like this method, there are a number of ways you can draw down carbon, and one is with crops, with agriculture, and the other is traditional engineering. You get a double win. You help farming, you help crops, you help the soil, and you also get carbon dioxide out of the atmosphere. You probably get a triple win, because until 30 years ago, when chemical methods got very powerful, a lot of drugs were found in plants. And if we have a big plant project, agrogenomics project, I'm sure that more pharmaceuticals will be developed. So there's a three-way win with this strategy. So what am I doing? I've got a very talented graduate student working on something, and this is different, called photorespiration. Now, the key enzyme in photosynthesis catalyzes carbon dioxide, of course, but it also catalyzes oxygen. When it catalyzes carbon dioxide, it does what it's supposed to do. When it catalyzes oxygen, it leads to a different pathway, a photorespiration pathway, which is poisonous to the plant. And the result is, and 80% of the plants on the planet have this pathway, this photorespiration pathway, and the other 20% have adapted. 80%, an important 80%, cereals, for example, all have this. So, and there's a loss of about 25% in yield as a result of photorespiration. The idea is to engineer the Rubisco enzyme so that the ratio of carboxylation to oxygenation increases. It's a hard problem. There are a lot of serious problems associated with it. A lot of people have got turned off from it for those reasons. They're fundamental. But I believe in the next 3 to 5 years, because of recent advances, it can be done. So Adam McCormick is working on this with me, and that's a subject for a much more extensive talk. And what we're doing with Gates, what we've identified as some very innovative technologies, and that, of course, is a subject for a different day. Thank you.
Okay, it's my prerogative to, as host, to get to go next so that I can just sit back and relax and enjoy everybody. So I'm just going to talk briefly about who's regulating the genome. So this is just an aspect of what we're working on in the lab. So oftentimes we think of a linear genomic sequence from whatever species as a series, the base is A, C, G, and T. But there's also the physical genome, right? In each of our cells, this 3 billion base pair long string is wrapped up tightly and is in complex with lots of different proteins. The major protein being the histone shown there in blue, and the DNA is wrapped around that. This histone can be modified, the DNA can be modified, but there's a whole host of other proteins that compact the genome and allow it to, at times, open up to turn on a gene. And so this physical genome is highly regulated. And my lab has been interested in who's doing the regulation and what are the proteins that do that, and how do we map them? So there are really two classes of proteins that I think do this and we're interested in. The first is shown in, does this work, the laser pointer? Shown in gray here. These are transcription factors. So they bind across the genome to little DNA sequences. So they say where in the genome you're going to do some function. And the other class is the cofactors. There's about 1,100 cofactors in the human genome. And they are the machines that do something. They modify the histones, they modify DNA, they move the nucleosomes around to open it up. They recruit the polymerase in to elicit transcription. And so there's an interplay between these two classes of proteins, cofactors and transcription factors. Now, both have been well studied, but what hasn't been well studied is who interacts with whom. In other words, this transcription factor, push the wrong button here, doesn't matter. The yellow cofactor is recruited by some set of transcription factors. And importantly, that can change between cells. It can change in disease states. It's highly dynamic. And so we really wanted to measure who recruits who, that interaction network between those two. So we've been developing this technique we've called Cascade. We just published our first paper. Here's what we do. We grow cells up, whatever cell it is. We take out the nucleus, get out all those regulators from the nucleus, and we put them on a small glass slide with hundreds of thousands of different DNA sequences, which we customize on it. We then, shown there in red, we apply an antibody to a cofactor of interest that has a fluorophore on it, and then we can scan it in a microwave, and we can measure the quantitative recruitment of that cofactor to thousands of different DNA sequences. I'm going to gloss over some details, but doing that, we can infer that red cofactor, which different transcription factors can bind you and pull you to DNA in that cell. And so we do that, and we kind of come up with these maps. And one of my students thinks of it as a fingerprint. It's a heat map. And in essence, what we're showing here is in each column is pairs of columns. There's cofactors that have been profiled as resting T cells and a stimulated T cell. And each bar indicates one of these complexes. It tells you, oh, that's the transcription factor or transcription factor family that recruits that cofactor in that cell at that time. And so what we're looking at is a panel here of 7 different enzymes called histone acetyltransferases. We were interested, this family do similar enzymatic processes, but we're like, well, who recruits which one? Do some recruit to promoters or enhancers? And so we can get this map. You'll see sometimes across conditions, the things don't change much. Sometimes you lose connections, sometimes you gain connections. And so we're using this biochemical map now to understand a few things. A, how does this predictor explain what's happening in the genome of a cell? How does this change in healthy versus disease states? How in cancer is this network rewired and altered? And so we're applying it to a lot of different biological systems and doing a lot of fun with it, developing the technology more, but we're looking for places to apply it. And so with that, I'll sit down and we'll talk afterwards. Thank you.
All right. Excellent. Well, thanks, Trevor, for inviting me. Yes, my name is Josh Campbell. I'm an associate professor down on the Med campus in the section of computational biomedicine. So I'm a bioinformatics and computational biology by training. In my lab studies cancer genomics. So we try to understand the mutations and copy number alterations that can cause cancer. And we study, one of the cancers we study is prostate cancer. And it turns out that there's a huge disparity in prostate cancer. So African-American men have worse clinical outcomes compared to other populations like European American men, including twofold higher mortality rate. And there's a lot of different causes to this that go into it, things related to socioeconomic status, access to healthcare, quality of healthcare. But there's also this question, are there underlying biological differences in the tumors of African Americans compared to other populations that may also contribute to this disparity? And so one of the things we want to do is look at the mutational profiles of prostate cancers from African American men to do this analysis, but it's really hard when you look at existing sequencing studies. So I'm showing you an example of a TCGA study. TCGA was a big consortium that profiled hundreds of thousands of tumors from different cancers, and they had a big study in prostate cancer. However, only 12% of men were African-American or Black in this study. And so a lot of these analyses, comparative analyses, were challenging. So one of the things that my colleague at UCSF, Franklin Huang, and I decided to do, along with my graduate student, Yusuke Koga, was to try to accumulate a bunch of different sequencing profiles from a bunch of different studies that each one had a small amount of African-American men, but by combining them all, we could get more power to detect differences. And so we accumulated data from 4 cohorts, and we looked at something, we looked at targeted mutation profile, or sorry, targeted sequencing data, and we had 15 genes in common, and a few of those genes, like ZFHX3, were significantly more frequently mutated in African-American men, and this was a novel finding that hadn't been reported before. And it was independent of Gleason score, which is a measure of aggressiveness of the tumor. We could also find novel tumor suppressor genes like ETB3, which were significantly deleted, but only in African-American men. So we only found this tumor suppressor by studying cohorts of African-American men that was previously missed in other cohorts. But just to be my sort of own critic here, weaknesses of that study were that African-American men and other groups were collected at different institutions. So they tended to be collected in academic, or white men tended to be collected in academic hospitals, Black men tended to be collected in other community hospitals. And so that could produce potential underlying confounding factors. And so more recently, to try to get around this and solve this problem, we've done an R01 where we're sequencing a bunch of tumors from Boston Medical Center and the UCSF VA. So we're gonna sequence 300 African-American men and have matched European American men from the same institutes, from the same clinical and socioeconomic backgrounds to be able to do that comparison in a better way. We've also gotten some pilot awards with a variety of people to do a whole genome sequencing in 25 American men and European American men. So it's funded by BU CTSI and Shipley and GSI. So those are all things in progress. And we hope to have data on that soon. Just for the last little bit, I guess before going on, what we're doing now, what I just mentioned was all sequencing on primary prostate cancer. So when you have localized disease, you can take the prostate out, and that's oftentimes curative. But prostate gets really nasty once it becomes metastatic. And it can metastasize to at various places like liver, lung, and brain. And the challenge with metastatic disease is that they don't oftentimes go in and take a biopsy of the metastatic site. So we don't have any tissue to do the actual sequencing and look at the mutational profiles. So one of the things we've been doing is studying circulating tumor DNA that can be found in the cell-free DNA. So you can actually take somebody's blood sample, isolate the plasma, sequence the plasma, and detect mutations and copy number profiles. So what's being shown over here are 4 samples taken from 1 patient. We use low-pass WGS to detect copy number profiles. This person came in starting to become resistant to androgen deprivation therapy. They were switched to a new therapy, and you can actually see that the amount of tumor DNA went down after they switched treatment. But after several more months, you can see the copy number profiles start to come back. And then at a 9 months time point, you can see very high tumor fraction in their cfDNA, along with many more copy number changes. So that tumor was sort of resistant to the treatment that they had just been switched on and had to be switched to a third treatment. And so from this type of data, we're hoping to understand the types of mutations that can cause resistance and how those may differ between populations, between African-American men and other groups. And this is a collaboration with people in Hemong, Gretchen Janak, and Matt Kolke. So I'll go ahead and stop there and pass it on.
Hi, everyone. Thanks, Trevor, for inviting me, and thanks, everyone, for coming. So I'm Juan Fuchsman Mas. I'm an assistant professor in biology. So different large-scale studies have identified millions of mutations in thousands of cancer patients by performing whole genome sequencing of tumor samples. More than 95% of the cancer mutations are actually located in non-coding regions of the genome, potentially affecting the function of regulatory regions. However, it is challenging to determine which of these millions of mutations are actually affecting cancer phenotypes. And the reason is because it's challenging to determine which ones are actually affecting the binding of transitional factors and cofactor complexes. Indeed, only around 70 known drivers have been identified in non-coding regions, although millions of mutations have been identified in patients. So the question now is, which of these non-coding mutations, of millions of non-coding mutations identified in patient samples, are actually cancer drivers? So traditionally, the method to identify or predict driver non-coding mutations in patient samples are burden tests. So this test, what they do is they find genomic regions that have an increased mutational frequency or burden across patient samples. And this is compared to a background mutational frequency that is estimated by comparing to different genomic regions with similar genomic features as the region of interest. The problem with this approach is that it's difficult to determine what is the expected background mutational frequency. And this has led to the identification of only few cancer drivers in non-coding regions. So what we did is we developed a variation of the burden test, which we call TF-aware burden test. And this was in a collaboration with Trevor here and with Adam Lavadorf. And the purpose of this burden test is to find mutations that share a common regulatory mechanism. For example, mutations that disrupt the binding site of a particular transition factor, and determine whether within a particular region, there are more mutations that affect the binding of that transition factor compared to the total number of mutations in that specific region and the probability that a random mutation in that region will disrupt the binding site of this transition factor. So in this case, instead of comparing to neighboring regions or to regions with similar genomic features, we're comparing to the region itself. So this leads to an increased statistical power and also increased confidence in our predictions. So using this approach, we predicted 2,555 driver non-coding mutations in the promoters of 813 protein coding genes, which is about 10 times more than what has been reported in the literature so far. These genes are associated with different functions, including housekeeping functions such as splicing, translation, vesicle transport, but also with cancer-related functions such as Wnt signaling, apoptosis, cell cycle, and p53 regulation. To determine whether these mutations actually affect transcriptional activity, we teamed with Ryan Tui from Jackson's lab and performed massively parallel reporter assays to determine the activity of thousands of mutations. So using this approach, we confirmed that around 800 of our predictions actually increase or decrease transcriptional activity. So this suggests that our TF-aware burden test can prioritize functional mutations. So the question now is, how do these mutations affect gene expression? As identifying the molecular mechanisms by which these mutations affect gene expression can lead to potential therapeutics. So mutations in non-coding regions can affect the recruitment of different TF cofactor complexes, of regulatory complexes. For example, mutation 1 leads to a loss of binding of this regulatory complex, whereas mutation 2 leads to gain of binding of another regulatory complex. Understanding which TF regulatory complex are affected by a particular mutation provides the opportunity for drugging non-coding variants, as there are many FDA-approved drugs that can target transcriptional cofactors. So to tackle this challenge, we teamed with Trevor to perform cascade experiments, which Trevor just explained a few minutes ago, to identify the transcriptional cofactor complexes that are affected by these mutations. So we tested the 2,555 mutations, and we identified 510 that alter cofactor recruitment, even when we only tested 6 of the around 500 cofactors that exist. So this suggests that we now have a potential mechanism for action for many of these non-coding mutations. And we can now zoom in, for example, to a particular promoter region, in this case of the EGR1 gene, which is a cancer-related gene. We can look at many of the different mutations that are found across different patients and determine which ones of these affect transcriptional activity. In this case, it's 6 of the mutations. Which ones are affecting the binding or the recruitment of different cofactors. And we can even compare different mutations if they share mechanisms, such as these 2, or have different mechanisms, such as this one here and this one here. And we can also identify the underlying transcription factors that are recruited by these different cofactors based on the motifs that are being affected. So in summary, we now have identified hundreds of cancer-driven mutations in non-coding regions and their mechanisms of actions, and we hope that this will inform future studies for developing new therapeutics. Thank you.
Okay, so this is my title. So you might be wondering what catching the drift means. And so here I'm referring to genetic drift. And we're taking advantage of the fact that nearly isogenic inbred strains, which are homozygous at every allele, through several generations will start to accumulate spontaneous mutations. And these mutations can have detrimental effects or changes in your favorite phenotype of interest. And so with the sequencing of the human genome 20 years ago or so, over 20 years ago, shortly thereafter, mouse genome sequences became readily available. And right around when I started my postdoc, I had this idea that I brought to my postdoc advisor. What if instead of using very different mouse strains and crossing them, if we can use very closely related mouse strains, might this facilitate our ability to map genes? And it turned out, yes, that's the case. And so basically what we can do is we can overcome the lack of recombination events in an F2 cross, because you only get 1 or 2 recombinations per generation, per chromosome, and take advantage of the fact that even though that we've identified these very large genomic regions because of LD, there's very few, in fact, 2 to 3 or 4 orders of magnitude, depending on the strain or if you're comparing to humans, fewer genetic variants underneath these loci that influence complex traits. And so with that said, this is the overview of my lab. We're interested in addiction. And so I'm a behavioral neuroscientist at heart. And so I'm interested in mouse behavior and specifically identifying and refining traits that we think are most relevant for understanding addiction. and also those that are most heritable. So ultimately, we want to apply genetic analysis to this. And so with these very closely related isogenic strains, we start with 2 genomes that are nearly genetically identical. Typically, 2 substrains is what we call them, differ by anywhere between 8 to 20,000 variants. That may sound like a lot, but any 2 classical inbred strains usually have about 5 or 6 million variants. Humans segregate, I don't know, somebody can probably answer this better, I'm guessing 30 or 40 million variants. So we're talking several orders of magnitude reduced complexity. So we cross these strains for a couple generations. We test them for our favorite phenotype of interest. We identify chromosomal regions, and then ultimately we combine this with gene expression analysis. So we map genomic regions that influence gene expression, overlay those with the influence behavior, and then identify candidate genes that we can target using genome editing technologies. When I first started, talons were the big thing. Now CRISPR, of course, is way cheaper to use, and so we're using that. And then ultimately, once we've validated a particular candidate gene, ideally a particular variant for our favorite behavior, ultimately we want to translate these findings to humans. So today I'm going to tell you a story where I think ultimately we hope it will translate to humans, not necessarily at the gene level, but at the mechanistic level. Okay, so what is this I'm talking about? So this is one of my favorite assays to use in mice. It's called conditioned place preference. So it's very simple in terms of how it works. On one side, you give them a saline injection. On the other side, you give them an oxy injection, oxycodone here in this case, oxycontin, oxycodone is the active ingredient. And we do this a couple of times. So we ask them initially what side they prefer. There's no bias for preference for a side. Pair them with a couple of oxy and saline injections, and then we give them a learning phase or consolidation phase. And then we test them either in the drug-free state. We just simply ask the mouse, what side of the box do you like better? And then on the next day, we give them a drug reminder. So we actually give them a drug injection. And what this will tend to do is that will activate the drug in memory, the CS-US association, conditioned stimulus, unconditioned stimulus. And the mice will make a beeline for the drug-paired side. So it serves as a reminder. And so what I'm showing you data is from these 2 very closely related strains, BALB/CJ and BALB/CDYJ. And what they show specifically is a difference in the state-dependent learning. So you give the drug as a reminder. And what you can see, the data is variable, but we'll get to the end of the story in a minute. You can see that in the females here, the female the female J's show a very large state-dependent preference for oxycodone compared to the very closely related J females. And so when we saw this phenotype, we thought, well, maybe oxycodone is just getting into the brain faster and it's serving as a more effective reminder for this rewarding memory. And so we measured oxycodone and its metabolites in the brain 30 minutes after an oxycodone injection. And you can see nicely, much less variable, so it's a more heritable trait. You can see that a nice increase, female specifically, in oxymorphone brain concentration. The oxymorphone is a metabolite of oxycodone. It's at least 10 times more potent and much more efficacious. And so long story short, we think this increased entry, this increased perhaps production in the brain of oxymorphone is serving as this reminder cue of how rewarding this thing. Oh, I'm already up. Oh, wow, okay. And so, so we mapped the QTL on chromosome 15 using a couple hundred markers to distinguish the 2 substrains. We combined this with gene expression QTL analysis. There was only one transcript that peaked within the same region as the region for oxymorphone concentration. And we've since conducted proteomic analysis, confirmed at the protein level that ZHX2 is a differentially expressed gene. And it's a candidate gene that we think is ultimately regulating the production of cytochrome P450 enzymes in the brain. Increased production of these cytochrome P450 enzymes leads to increase in oxymorphone brain concentration and an increase in these drug reward related behaviors. And so we're testing this hypothesis by removing the mutation, which is a 6KB MERV, and then we're also doing the opposite experiment where we're removing exon 3 to mimic the MERV and seeing if we can recapitulate the phenotype to show necessity and sufficiency. Sorry I went over, that went really fast. Okay, thank you.
Hi, I'm Christopher Schmidt. I am not on the medical campus. I'm here on the Charles River campus. I'm a biological anthropologist, so I'm also a little different in that respect. That means that I really study these phenotypes, both biomedical and various others, primarily to understand a little bit more about human evolutionary history. And in my particular case, I'm mostly interested in understanding the evolutionary history of metabolic disorders. So I did start out on biomedical campus, and then I ran away back to anthropology because I'm more comfortable there. You might see why in a moment. Since I'm interested in metabolic disorders, I understand, and I'm sorry for anybody who studies humans in relation to these, but they are terrible for studying metabolic disorders. We lie about what we eat. We lie about how much we exercise. We can't just follow them around and track everything they're doing, although people have tried, but NIH funding horizons are like 5 years, so it doesn't really work developmentally. So I thought, what about monkeys? And my actual training is as a primatologist. I started out in behavioral ecology, although I was doing some Sanger sequencing and microsats on the side to take it back a ways. And in my first postdoc, I got involved at the ground floor with the International Vervet Research Consortium, which was really trying to set up a model system for creating a comprehensive way to really go from genotype to phenotype in very complex traits. In particular, as a way of bridging kind of the biomedical approach towards understanding phenotype-genotype interactions and really being able to branch it out into having a more evolutionary understanding for how these traits develop. So the way that we did that is they hired me to trap thousands of monkeys in Africa and in the Caribbean. You can see kind of the system here. So I did go out to the wild. This is an image of me trapping with some BU students. So I do occasionally bring them along. That is a monkey about to be trapped. Very, very simple way of actually collecting these data. And so we built up a resource where we've got around 3,000 monkeys that we've trapped in the wild to capture kind of wild phenotypic and genotypic variation. We trapped on the islands of St. Kitts and Nevis, where they were brought over about 400 years ago. So they're very kind of inbred. There's a bottleneck. So there's like really nice long linkage blocks to associate genotypes with phenotypes more simply than we can do in the wild, where there's a little too much variation and noise. And then we also have a captive colony, the Vervet Research Colony, which used to be at UCLA, but is now at Wake Forest in North Carolina. And in that colony, they're pedigreed. We've got about 950 animals pedigreed. 720 of those are whole genome sequenced, which really gives us good power to use both this pedigree and the genomes to really make those linkages between the genome and complex phenotypes. And so when I really was done trapping these monkeys, they're like, we don't know what to do with you. You're an anthropologist. That's weird. They decided to send me to this colony. And as I already mentioned, I'm interested in metabolism and development. So I was like, okay, well, some of these monkeys, they're becoming obese. Why don't we see how that actually develops over time? And because they had been pedigreed and measured, and every kind of 3 months they have to be taken down and tested for TB, during which time they're also measured, this gave us really good resolution for tracking developmental patterns related to obesity that give us really nice images of what these traits look like over developmental time. On top of that, because they're all pedigreed and because we've got their genomes for the most part, we could actually make associations between those developmental patterns and variation that was in the genome there. On top of that, since they're not human and we can actually watch everything they're eating, we also found out that there were some really interesting kind of maternal effects. This bottom graph, I'm not going to explain it in detail, but on the far left, what you can see is that animals that were fed a very specific kind of challenge diet, when their mothers were eating that diet, they actually were more likely to be obese once they reached adulthood. So there's a really nice kind of story in the captive colony of obesity developing over developmental time, but on top of that being influenced by various environmental factors that we can test out. Now, with this large system that we've built up, we also can go from mapping out regions of the genome in that captive colony that are associated with metabolic disorders. We do that through multi-point linkage using the pedigree to increase our statistical power. And then we can go back to the wild where we've whole genome sequenced about 163 animals and actually kind of trace that variation throughout the wild population in Africa. In that way, we can actually trace these variants across different environmental gradients, find out how those populations differ, and then use various tests to actually link those traits to selection in relation to ecogeographic variables. So all of these figures are from my PhD student Christian's first dissertation chapter, pointing out in UCP1, which is one of the genes that we found in captivity associated with obesity, it actually varies in the wild and is actually under selection in Southern Africa. Those monkeys that experience really cold winters, they actually show signs of selection within that regulatory regions for that gene. So we're really making that link between captive obesity and wild variation and selection. Now, the next steps for this, I have PhD students continuing with the vervet model. We're looking at microbiome across farm and wild sites to see if diet is actually shifting. And it does look like those farm monkeys look more like captive monkeys and how their microbiome's responding. I've got funding to eventually go back post-pandemic and look at gene expression across various conditions, including drought, to test out hypotheses for the development of obesity. And I'm also using portable genomic technologies in Peru to build another model system in a critically endangered primate to understand how they adapt to various ecological conditions that they're forced into because of deforestation and hunting. So really kind of nice buildup of a system relating, again, these biomedical concerns to more of an evolutionary frame. Thank you.
Okay. Hi, I'm Sarah. I'm an assistant professor in the Department of Biology here at BU. I've been here since 2017, and I'm going to talk to you not about humans or anything with a vertebrae. I'm going to talk to you about corals and climate change. So actually, Charles led nicely into what I'm thinking about in my lab. So we think about the effects of climate change and how they impact coral reefs. So I don't hope I need to introduce anyone to climate change. It's warming our oceans. As Charles mentioned, up to 90% of the CO2 emitted in the atmosphere is actually absorbed into the ocean. And let's see, I did some animations on accident. Okay, so we're really interested in corals, and corals have this really unique symbiotic relationship with dinoflagellate algae that actually live intracellularly. So they somehow allow algae to like live inside their cells. Coral reefs are really fascinating because they exist across this really wildly different environmental landscape. So if you imagine, this is just zooming on the Florida Keys. This is looking at seasonal variation, but there's also like daily variation. There's like reef zone differences. And if you're closer to shore, you're experiencing more anthropogenic like runoff and things like that. But you know, we usually think about this relationship of the coral with its algal symbiont that creates this like dysbiosis where they exhibit this bleaching phenotypes. That's where they lose their algae. But they also have this really fascinating symbiotic relationship with this microbiome. So really it's this tripartite relationship. We think of it as like the holobiont. So this level of complexity of the holobiont coupled with the complexity of the habitat makes it really challenging to predict coral bleaching in a context of conservation. So, oh yeah, that's the coral holobiont. I'm just gonna make sure this is all in here. There we go. Okay, so when we think about this, one of the main reasons why it's really challenging to predict coral bleaching is that there's just a ton of genetic variation. So on the left here, this is looking at some corals. I should also mention that the first coral genome wasn't even sequenced until like 2015. So we're like over a decade or more behind things like human genomes. And we also have like way fewer resources. So just like a caveat. So this is some work we've done in Bocas del Toro in Panama. And we found, this is just an admixture plot looking at just shallow, we use RADseq, which is just like resequences about 1% of the genome. But you can see there's like really strong assignment into 3 different lineages. These lineages partition really differentially across the seascape, with some lineages being more common inshore and ones being more common offshore. When we did thermal challenge experiments on this work, we found that one lineage, this deep purple lineage, which actually exists offshore, performed much better. This is just our metric of thermal stress response, whereas this lineage kind of like, well, slowly died essentially. And all this work was done by Hannah Eichelman, who's a PhD student in my lab. We also have seen the exact same thing. Now I'm going to move you into Palau, so into the Pacific Ocean, and we've seen the same thing. It's wild. So this is work done by my postdoc Cass. We see again like 3 to 4 cryptic lineages. They are differentially partitioned across the seascape, and they exhibit differences in thermal tolerance in response to thermal stress. So this is like wild that we see the same thing across the seascape. But this amount of genetic variation, that's all cryptic. So all these corals, even for a coral biologist, they all look the same. So it's like not like you can go do experiments and say like, oh, this population is more thermal tolerant than the other. You actually have to sequence their genomes to actually know who they are first. So this is one reason why we think it's really hard to predict coral bleaching. The other reason is for this. So this is, so we also work on these facultative corals, which are like really rare. There's only 8 species in the world, but they exhibit this really neat relationship with their algae where it's not obligate. So the corals I told you about before all have to have their algae. When they bleach, they essentially die of starvation. This species can live with or without their symbionts, and you can also culture their symbionts. So you can do experiments of the host and the symbiont with and without each other. So it's really powerful. And we use a lot of gene expression as like a metric of how you respond to stress. So this is just some work actually done by undergrads in my class, where we put them under cold challenge and heat challenge. And this is looking at relative gene expression plasticity in response to these stressors. Okay, I'm just going to tell you the main points. So this is showing that the host symbiosis leads to stronger response in heat. So we see that when the host is in symbiosis, it has a stronger response. When we did the same kind of experiment on the algae, we saw that it was the opposite. So when algae worked in symbiosis, they exhibited stronger responses. So there's these really weird ways that corals and their algae are responding to stress. Oh yeah, that's Dan and that's Hannah. That's who did the work. I don't know why this is annotated. Okay. The future work we're doing is we're doing a bunch of whole genome resequencing in Palau and also some work in Morea for like an actual in situ bleaching event. And we're doing whole genome resequencing to try to get at the genomic architecture bleaching. And then we're also pioneering some single cell RNA-seq in this model that we can actually separate the symbiotic relationship in the lab. And this is work with Maria. And we're looking at a lot of NF-kappa B transcription factor work with Tom Gilmore with an NSF we have. And we're also doing a little bit of work on this little dude here, which doesn't calcify, but it exhibits this facultative symbiosis too. So hopefully we'll know more about symbiosis, but probably a lot less than we know about cancer.
Hey everyone, I'm going to very quickly tell you whether cold sores cause Alzheimer's disease. This idea has been around for a long time that microbial infections, either through chronic inflammation or direct neuronal damage, might be risk factors for dementia when we get older. Amyloid beta itself has a lot of immune-related functions. If you inject virus into a mouse brain, it starts expressing amyloid. They've actually isolated human herpes virus from human amyloid plaques. And most recently, another group used RNA-seq data to show that the quantity of viral RNA in brain samples is associated with Alzheimer's disease risk. So we thought if we could do it with RNA, why not we do it with DNA? So I have access to a cohort of about 40,000 Alzheimer's disease cases and controls, primarily taken from blood samples, but several thousand have brain samples and split approximately evenly between whole exome and whole genome sequencing experiments. So we thought, hey, can we detect virus in there? And the whole reason any of this works is because of the nature of certain infections like herpes, as opposed to acute infections that you get and then clear. Herpes has this insidious ability to, crawl up your neurons and hang out dormant in your brain only to reemerge when you have wedding pictures or a first date. And so what we do is basically take the junk that's left over from human sequencing experiments that don't map to the human genome. We try to map them to viral genomes. How sequencing experiments work: you take your sample, you grind it up, you isolate the DNA, you chop it up, you sequence it, and you map it back. So whatever DNA is in there gets sequenced. DNA is DNA, whether it comes from a human or a virus or an axolotl. So we did that, and we really don't know whether this is going to actually detect virus or whether it's just junk. But for several reasons, we think we actually are detecting actual virus. First of all, we detected about 60 unique species across all the different strata of the data. We detected many more in blood, which is what we would have expected to see. Some viruses can't get into your brain. 10 viruses were detected in brain, blood, whole genome, whole exome. Most importantly, I think we only detected virus at all in about 40 to 50% of the samples, depending on the which strata. So, if it was just all sequencing artifacts, we'd probably find them in everybody. And what's really nice is that the most common viruses we detect, turns out, are the most common ones that affect humans. By far, the most common one we found were from the herpes virus family. Here's the families. Human alpha herpes virus 1, which is what causes cold sores, was by far the most common. And the second most common was human papillomaviruses, which are known to be very common. And then finally, these ones called torctenoviruses, which not a lot is known about them. They're not known to cause any disease, but we found a lot of them. And then also, these are the blood samples, whole exome and whole genome. We see the same pattern, whether it's blood, brain, whole exome, or whole genome, the same patterns
are observed. We detecting the same viruses most commonly. So we take these viruses, we count them up. Our PhD student dumps them into a bunch of different machine learning models. We determine how many of the models determine that virus to be a significant predictor of AD. And turns out we see lots of different human herpes viruses. We see a lot of human papillomaviruses and a lot of these tortenoviruses coming up as significant predictors. So then we take those, we put them into normal regression models to get effect sizes and things like that. It turns out after correcting for multiple tests, we see 3 viruses that were significant predictors of AD, HSV1, HPV71, and this tortenovirus 10. And that's about it. Our future goals, we're looking at whether there are genetic risk factors for both viral infections themselves or whether they interact with viruses to predict AD. And we're also looking at some endophenotypes and neuropath and some biomarker data to see if we can get at some of the mechanisms through which these things might be affecting AD risk. And I think I finished early.
Okay, last one. Thank you for staying and for the invitation. I'm Rachel Flynn. I'm an associate professor in the Department of Pharmacology. Back to the med campus for a minute to talk more about cancer. My lab really operates at the intersection between DNA damage repair and telomere biology. I've had really a longstanding interest in pediatric oncology and specifically in osteosarcoma. And so one of the hallmarks of cancer cells is the acquisition or bypass of replicative senescence and the acquisition of cellular immortality. And of course, we can attribute this back to mechanisms of telomere maintenance. We know that as cell divides, telomeres erode. That erosion ultimately triggers a DNA damage response that causes the cell to stop growing and ultimately either senesce or induce apoptosis. And so cancer cells have to overcome this by activating mechanisms of telomere maintenance. And so one of those mechanisms is reactivation of the enzyme telomerase. So reverse transcription is recruited to the end of the telomere and promotes DNA synthesis and extension. And so this happens in all cancers, and in osteosarcoma, this happens to happen in about 40% of all osteosarcoma. If you look specifically at pediatric osteosarcoma, this number decreases even more. About 20% of pediatric osteosarcomas rely on reactivation of telomerase for telomere maintenance. The other mechanism, which maybe people have not heard of, is this mechanism called the alternative lengthening of telomeres pathway. When I was putting this talk together, I said it was going to take me 4 minutes just to get that title of the pathway out, but this mechanism is not as simple as telomerase. It's not a simple reactivation of an enzyme. It's a very complicated pathway, and it's based on homologous recombination, a mechanism of DNA damage repair. The premise for the activation of this pathway, the idea being that there are significant alterations in the chromatin landscape, specifically at the telomere, that lead to loss of heterochromatin. This causes a more permissive environment that allows for recombination at the telomere end. And so what happens in these instances is that a critically short telomere will actually invade a neighboring telomere, and it'll use that sequence as a template for DNA synthesis, telomere elongation, and the acquisition of immortality. And so currently, there are clinical trials for the testing inhibitors of telomerase. There are no treatments or inhibitors for the inhibition of the ALT pathway. And so for cancers like osteosarcoma, and especially ones in children, right, there are going to be no great targeted therapies unless we can therapeutically intervene in the instance of ALT. And so how do we go about doing this? I would say that the significant limitations as it stands now are that we know very little about the genetics of ALT, especially in osteosarcoma, and we know very little about mechanistically who are the players that are involved in this pathway. And so to get at this from sort of both those angles, we initiated a collaboration with a clinical investigator, Dr. Peter Houghton, who's at Nationwide Children's Hospital. He's part of the National Cancer Institute's Pediatric Preclinical Testing Program. They're able to isolate osteosarcomas from patients. They send them to us. We're able to chop them up into fragments. We can either preserve them or implant them into mice. We can take the tumor that's grown in that mouse. We can excise it, fragment it, and then serial implant it into additional mice. This allows us to generate more material so we can characterize ALT. First step being, is this an ALT tumor or not? I would explain how we do that, but again, it'll take me 20 minutes to do so. So you can take my word for it. We can characterize ALT in the lab using a lot of molecular tools. We can then take that material and do some genomics on it and analyze, are these tumors, do they have mutations in some of the players that are involved in DNA damage repair? And ultimately, will that guide us in identifying genetic vulnerabilities that we can ultimately target therapeutically? And so the ultimate goal being, can we take this from the genetics down to models in vitro or in vivo, and again, look for identifying tractable targets and treatment. And so how do we go about doing this? We started doing this initially. I think we've analyzed about 30 tumors thus far. And this has really been a team effort. We've had some collaborators here in the room. Dr. Adam Labradorf has really been instrumental in providing some computational support to identify the genetics of these tumors. So in some of these samples, I would say to date, we've identified 3 novel mutations that had been previously uncharacterized and also had not been contributed to mechanisms of alt. And in this context, I'm just giving one vignette here, where using sequencing, we identified a translocation between chromosome 6, which probably is hard to tell, and I don't have a pointer. Oh, here we go. Chromosome 6 here and chromosome 16 here. And in this circles plot, we can see that those 2 pieces of DNA have been joined together to create a fusion between this protein DAX and this protein KIF-C3. What happens in this instance is this actually leads to a chimeric protein that functionally inactivates DAX. This leads to loss of heterochromatin formation at the telomere and helps activate this alt pathway. We can visualize this in more ways than just sequencing. So using an orthogonal approach, we use spectral karyotyping. So in these experiments, it's a metaphase spread of chromosomes from this tumor. You can see that instead of having 2 copies of every chromosome like it should, it has in some instances 4, in some instances none. In spectral karyotyping, the chromosomes are all painted a single color with fluorescently tagged probes. They should be at least the same color. And what you can see from this is if we look at chromosome 6 here, you can see a pattern of pink and blue. This actually sits at that fusion site between DAX and KIF-C3. So it's highlighting the translocation in these tumors. And if you look at the other chromosomes, you can see you get this, this striped pattern indicating that these tumors are really incredibly genomically unstable in general. And so what do we do with this information? Well, as I mentioned, the ultimate goal is to identify tractable therapeutic targets in the lab. And so one of the things that we have identified in the lab is that these cells are actually incredibly dependent on a DNA damage response kinase called ATR. And so in taking a number of cells in culture, primary cell culture, we can treat these cells with an inhibitor of ATR, VE821. We can calculate the IC50s from that treatment, which are plotted here. And we can see that the IC50s in alt positive cells in blue are significantly lower than the IC50s of cells treated with ATR inhibitor that are non-alt, and most of which being telomerase positive. This is unique to ATR, as treatment with an inhibitor to another DNA damage response kinase, ATM, does not stratify alt positive versus negative. So these are in cell lines. Ultimately, we want to test those in those patient-derived xenograft models I described earlier. So this is not the actual experiment, but in fact, just the growth of those tumors that we got from the preclinical testing program, where each color represents a different mouse with a tumor growth that we can then ultimately validate the preclinically these studies that we've done in vitro. Thank you.